Hi everyone, I'm Xinying Zhang. I'm a senior undergraduate student from Memorial Shanghai. I am majoring in both neuroscience and chemistry. My DERF project is training induced hippocampal synaptic maturation in rats. It is my greatest honor to have the chance to work in Albrini lab, and I really appreciate all the postdocs, graduate students, and our lab manager and our lab for supporting me, helping me out, and teaching me throughout this whole project. I would like to thank to our principal investigator, Dr. Christina Albrini, who is also the faculty member of my DERF project. I started this project at the beginning of June. We are interested in determining biological changes induced by learning in the hippocampus of rats trained at postnatal day 17. It is a part of our larger goal of understanding biological mechanisms underlying long-term memory formation in rat pups. We are interested in how the long-term memory is formed and stored during infantile learning because there is a phenomenon known as infantile amnesia, which is the episodic memories formed during the first postnatal period are rapidly forgotten. But interestingly, previous work in our lab shows that the memory during the infantile amnesia period are not lost and can be retained by reminder later in life. Also, the infantile memories are stored using the mechanisms involving the dorsal hippocampus. Furthermore, training during this period increases the markers of excitatory synapses in dorsal hippocampus that last for at least 48 hours after receiving the training. Earlier in our lab, our Western blot experiments has shown the results that the synaptophysin and PSC95 are both increased for trained PN17 rats. So these proteins are synaptic markers that we used. Synaptophysin is a presynaptic marker and PSD95 is a postsynaptic marker. So we would like to use immunohistochemistry as an approach to really look at the images of the brain tissue. The training we give the rats is inhibitory avoidance task. The rats stayed in a chamber made up by two compartments, one bright and one dark. The rats would prefer to enter into the dark compartment. When they entered, they would experience a foot shock through the grid floor. Thus the memory like, "This room is not a nice room" would form. And when they are tested, they would be reluctant to enter into the dark compartment. In our experiment, we have naive, trained, and shock-only rats. Each condition has 3 samples. The brains are cut into 30 micrometer slices. We picked up slices that were pretty much the same to one another for 3 conditions. The slices were incubated with primary antibodies for 48 hours under 4 degrees and then developed with secondary antibodies. This is our microscope room. We can book the microscopes here for taking images to analyze and to check our preparations. The fluorescence groups on the secondary antibody will shine under the laser with corresponding wavelengths. Here is the Leica confocal microscope. It is a microscope with very high resolution, under 63x with the refraction of oil that we applied on the slice. We will be able to see fine signals like the synapses that we are interested in. We use it to take images that we can use for quantification. This is the VS120 microscope. It can only support 40x resolution at maximum. Therefore, our puncta will appear only like the stars we see in the sky at night. We only use it to look at our preparation, meaning to check if the staining worked or not. But it is still a useful microscope because it scans much faster and can quickly give us a general idea before we spend much time on the confocal. Here is one of the several confocal images that we obtained. The red punctus showed the presynapses with synaptophysing label, and the green ones show the PSD95 labeled postsynapses. Some of them are very next to each other, and some are even overlapping. These are the co-localized synapses. We have quantified both synaptophysing and PSD95, as well as the colocalization. The results have shown increase of them. For the further study, we are going to improve our way of quantification. We are also going to use another approach, which is the high-resolution confocal laser scanning microscope and Neuron Studio spine morphological analysis. It can create a 3D picture that reveals what the condition of the synapses truly look like in the specific region of the samples. I truly appreciate the DERF program and Alberni Lab for having me this summer because I really learned a lot. It is not just about doing experiments. I mean, it is an important part in the sense of practicing skills, learning how to use variable equipments and softwares, etc. But I feel that the most valuable thing that I get from it is the sense of doing research. Starting from finding a specific angle and a large goal and write the proposal about it, planning everything accordingly and overcoming various troubles during the experiments, and to eventually get a result from that. And getting an experimental result is actually not a finale that we would use to think about. The further interpretation of the experiment result is actually a more important part, I would say, because how to interpret the data and the result will lead us to think further about the question, and that is what makes up the discussion part and eventually would make people to think about a further question that arises from the project. The hardest thing I encountered during this project is about setting the conditions. It took a long time for us to find the right antibodies to do the double staining. For about a month, we kept doing the condition testing to stain both synaptophysing and PSD95 on the same slice, but sometimes the PSD95 antibody didn't give the signals, or sometimes the dilution was not appropriate, but we have eventually got the appropriate condition and applied it to the actual experiment. It is also a lesson to me that science is something that you need to keep trying and find the solution by yourself. There will not be someone that have everything said and you just need to follow others' path. It's a discovery. That is the charm of science.